Good morning. I'm Sujata, and these are my teammates Aishwarya and Fauzia. So today we are here to present a video on our project, which is development of hydrogel infused organic fertilizers for sustainable agriculture, under the guidance of Dr. G. Vijayalakshmi Ma'am, Associate Professor of CBIT. These are the contents of our project. Coming to the introduction, climate change, resource depletion, and environmental problems such as global warming, etc., threaten our future. Agriculture gives us food, materials, and jobs, but due to these problems I've stated above earlier, like shortages and increasing population, there are various other problems arising in agriculture. We can help feeding the crops gradually by reducing the waste and our project main motto comes where we are trying to plan a sustained release of fertilizer, which makes the soil better, and hydrogel, which slowly gives the plants the water in helping in dry areas by saving the water. Vermiwash helps the plants to grow and stay healthy. So by combination of these both, that is the vermiwash and hydrogel, it can give better growth for the plants by improving the soil health. Coming to the literature review, as we know, globally, agriculture is the greatest consumer of water, and survival of the plant completely depends on the irrigation. So, this requires a better and improved water management system, which is successfully provided by the hydrogens. So, hydrogels, the basic motto behind the usage of hydrogels in agriculture is mostly improvement of water management because when we are using hydrogels for agriculture, there is no need for regular irrigation. And next, enhanced nutrient delivery, where hydrogel can encapsulate and slowly release the fertilizer or pesticide, whatever the nutrients we are thinking of producing to the plants for the growth. And it optimizes the nutrient use efficiently, minimizing the environmental pollution. And the other thing is it is environmentally friendly because it is biodegradable and non-toxic nature. And the usage of hydrogels also acts as soil conditioners because it improves the soil structure and aeration by increasing the water holding capacity. And last but not the least, stress mitigation is also increased where the hydrogel helps the plants to withstand extreme conditions like drought, heat, and salinity. Coming to the next one, where we are using CMC for as a natural polymer for hydrogel over other biopolymer based hydrogels for controlled release of fertilizer because of its high thermostability, and CMC has 100 times more water holding capacity than any other biopolymer, and it is non-toxic in nature, hence it doesn't cause any adverse effects. Next, coming to the organic fertilizer, which we have preferred of using, which is vermiwash. Vermiwash is basically extracted from the vermicompost, where vermicompost is kept on a vermi bed and the water is run off. And the water runoff is the vermiwash, which has higher concentration of nitrogen, phosphorus, and potassium, which is required for the plant growth. And due to this water runoff, the carbon content actually decreases in the vermiwash. And the reason why we use vermiwash over vermicompost is as when we are trying to encapsulate vermiwash with the hydrogel, vermicompost is not an ideal thing because vermicompost doesn't have stress mitigation properties compared to the hydrogel. So if we have to incorporate vermiwash with the hydrogel, it is the better to use vermiwash as the organic fertilizer. Over to Aishwarya.
So based on these literature reviews, we formulated our aim to develop an organic fertilizer by infusing vermiwash into CMC-based hydrogel. Our main objectives for this experiment was hydrogel development and characterization, plant growth experiment and its comparison, and qualitative analysis of vermiwash and the soil used. Next page. The novelty of this project is this is for the first time to infuse vermiwash, organic fertilizer, into hydrogel where specific desired characteristics such as water retention capacity, and slow nutrient release of hydrogel, and increase and nutritional value, and increasing soil health by vermiwash. Uh, a combination of both these properties would uh, ultimately result in a good fertilizer, that too in an eco-friendly and cost-effective way. Next slide. So to proceed with the aim and the objectives, we designed our work in few phases, which includes and uh, preparation of hydrogel, infusing it with vermiwash, and comparative studies and plant growth experiment, and the qualitative analysis of soil and vermiwash used to know the components present.
So in the procedure, as we have discussed in the workflow, so on preparation of hydrogen infused vermiwash, you need 10 ml of vermiwash, and we have added 90 ml of distilled water. We have used different concentration of CMC plus 10% of citric acid to provide the best CMC for hydrogen infused vermiwash. And we have added the CMC into the vermiwash and on hot magnetic stirrer, and we allow it to sit for 24 hours and to remove all the air bubbles. We then cast the hydrogel solution and put it in hot air oven at 60 degrees centigrade. And we have obtained a vermiwash loaded CMC film. We have even tried with bead formation 2 m of calcium chloride, but but the hydrogen film was not as accurate as you have seen in the previous film. Next. So we have made the powdered hydrogel. In this experiment, we have added 50 grams of soil. Layer by layer technique is used, in which we have added 50 grams of soil on top of the 0.75 grams of hydrogel and covered it with 50 grams of soil. You can see the three samples: controlled, vermiwash, and hydrogel. Next. So in the first result, we did not use a technique of layer by layer. We have mixed the hydrogel with the soil, to which the water could not percolate in hydrogen infused with vermiwash soil. As you can see, the better results were with vermiwash and control. And this is the layer by layer technique, where we have cultivated fenugreek and coriander seeds. Hydrogel seeds germinated in just in 2 days, whereas vermiwash and control were around 4 to 6 days. But the stability was seen more in hydrogen based infused vermiwash than compared to vermiwash and control. So we have planted 20 seeds, out of which we have gotten better results with hydrogen infused vermiwash, which was around 18 seeds, and the shoot length was measured around 0.16 to 2.4, and the germination of seeds was seen in less than 2 days.
Further, we extended our procedure by growing replicates by planting 40 seeds of mustard and fenugreek to assess how fertilizer developed is impacting soil health and morphological features. Next. Next slide. So, as we can see in the picture, the plants which are treated with hydrogel, as usual, showed better result. Next slide. So, plants treated with hydrogel infused vermiwash showed 80% of seed germination with 16 days stability and 13.3 centimeters shoot length on an average. Wherein vermiwash treated plants showed 65% less growth than hydrogel ones with 12 days stability, and plants which were took as control, that is treated with nothing, showed less germination rate and stability of plants. Uh, the, by this, we can conclude with that organic fertilizer developed worked well. Next slide. Now, to know how the fertilizer developed enhance soil health, we proceeded with fluorescence test of vermiwash, and this test ended in positive result showing fluorescence in vermiwash. To know regarding but as of our knowledge, no literature review explains presence of fluorescence particle in vermiwash. Then, to know the components present, we tested it under UV spectrophotometer, which depicted a slope in the graph. So, uh, for further we have gone through FTIR analysis of this vermiwash as well as the soil, and to know whether the and to know how the components are affecting soil health and uh, nutritional value of the plant.
Coming to the FTIR analysis, which is Fourier transfer infrared spectroscopy. So as we know that vermicompost or vermiwash basically is prepared by usage of various waste and which is treated with the earthworms. So basically it has higher content of vermin. So the main functional groups in these are OH and CH2. So as we can see here, this is the comparison of hydrogel and vermiwash. So where we are doing this to confirm whether there are vermiwash components incorporated into the hydrogel or not. So we can see the similar peaks near the wavelength 3400 to 3200 range, and as well as near 1600 range, and as well as between 1200 to 1000 range, which actually proves that there have been vermiwash components which are being incorporated into the hydrogel. So the hydrogel actually consists of the vermiwash contents. And coming to the next one, here we have taken the soil samples where we have compared the hydrogel soil as well as normal soil to compare the peaks whether the hydrogel contents have been incorporated into the soil for the growth of the plants, where we can see the similar peaks between 3400 to 3200 and near the 1600, as well as from the 1200 to 1000. Here we can see that the hydrogel soil and the normal soil difference peaks. We can see that the components of the vermiwash have been incorporated into the soil, showing more effective vermiwash components in the soil, which cause greater soil health and plant growth. So these are the wavelength numbers and peak types, where the components and functional groups that have been found in the vermiwash and the soil components and hydrogen. Over to Fauzi.
Okay. So our next step was to determine the color of the leaves, that is the percentage of chlorophyll. As we all know, chlorophyll plays a crucial role in the process of photosynthesis by absorbing light energy and converting into chemical energy. For this process, we have taken the leaves of mustard, of control and hydrogel, and we have sun-dried and grinded into fine powder, to which we have added 0.1 gram of powdered plant leaves by taking in 50 ml of beaker. 30 ml of acetone was added at 4 degrees into the beaker. 3 samples were taken, which is the blank, hydrogel and control. For the sample, only 30 ml of acetone was added, but for the hydrogel and control, we have added the dried plant leaves, and we have measured the absorbance at 645 and 663. As we know, the absorption spectrum of chlorophyll includes wavelength of blue and orange red light, which is around 450 to 475 nanometers and around 650 to 675 nanometers. As you can see, the chlorophyll extraction in hydrogel, it is more greener than compared to the control plant. Next. So in the chlorophyll test, as I've said, 3 samples, blank, hydrogen, control, and the average was seen more in hydrogen than compared to the control, and the chlorophyll, as we have seen, the chlorophyll test was performed. So the total chlorophyll content. So there are majorly 2 types of chlorophyll, that is the chlorophyll A and chlorophyll B. Chlorophyll A, it is the abundant type of chlorophyll which absorbs the light rays of wavelength. And chlorophyll B is a pigment which is responsible for passing on the light energy to chlorophyll A. and the total chlorophyll content for A and B was seen much higher, which is 1.5x greater than that of control. Next. So expected results. As we have done this experiment based on lab-based, the results what we have seen was the cost-effective method, longer stability to plants, and to control of leaching by water. But on a large scale, when we have to use this hydrogel infused vermiwash in agriculture basis, we want these results to be expected. That is the long-term nutrient availability, improved crop productivity, improved soil health, reduced nutrient runoff, mitigation of drought stress. Thank you.